Hello, sir. Good evening.
Yeah, good evening, Majid. Just give me 2 minutes, I'll join you. I'd like to have a cup of coffee.
Yes, sir, sure.
Hi, Vaibhav.
Hi, sir.
Yeah, you regarding your 510K, you got it resolved, or should I explain more?
Sir, that's not my issue.
What?
So that was Aman, I guess.
Acha, sorry.
Yeah.
I got confused because Aman also keeps asking. I mean, Vaibhav also there. Aman keeps asking a lot of questions, so I got confused. So whoever it is, yeah, Aman was in dentals, no? Dental implants and dental composites. And Majid, what is your product? What are your products?
So they are ENT medical devices plus hemostats, wound care management products.
Acha. Yeah, I know this. They are all hemostats, no?
Hmm.
Like, yeah,
Yeah,
gelatin based.
hemostats, gelatin, ORC, cellulose, chitosan, um, PVA. lots of wound care based
Yeah,
alginate and
yeah.
calciums as well.
I work
So we
for one of
make
the company on
absorbable.
hemostats.
Mhm.
Like Aegis Life Sciences was their Axio Biosolutions in Abra. Both of them work on these hemostats only. But Axio Solutions, they have, I think, it is chitin based or something. But
Most probably, yeah, chitin based entire portfolio. AGs have more into gelatin and ORC, I guess.
yeah, Aegis was in, I mean, Aegis bread and butter is gelatin.
Oh,
Okay, ORC, I mean, they are importing from South Korean company called Samyang.
Samyang. Yeah, I'm
Yeah,
aware about that.
even
Most
PV.
most of the companies here in Gujarat they export from Samyang, I guess.
Yeah, yeah.
Yeah,
Because
I
I
took a project
audited.
for Merrill back in 2024 for their ORC. So
Yeah,
right now,
actually, the point is like very most, I mean, ORC, most of them are exporting it as sheets and they're
mm,
doing some preliminary work like, but I have worked, when I work, like I have a project where I am doing,
mhm.
where, yeah, from cellulose we can prepare ORC, like oxidized
I
regenerated
have done
cellulose.
that for Merrill actually. For the ORC, they gave me one. They hired me as a consultant for certain 6 to 7 months, and for them, I made ORC for by doing oxidation reaction. So we use certain inert material together with NO, NO2 liquid, NO3 liquid, and
Right.
with that we did oxidation and prepared that ORC. I'm not sure whether they opted for the large scale production or not, but for pilot scale they hired me once.
No, no, no. At large scale, they are still getting it from Aegis Life Sciences only.
Oh, okay.
Aegis is a supplier, I mean, OBL for Merrill.
Acha, is it so?
Uh, and
Hmm.
uh, and also for Surgy Spoon, that gelatin sponges, no?
Mm-hmm.
I mean, even Merrill is also getting this so on OBL. See, PVA, I don't think the synthetic hemostats, except for nasal and ophthalmic, they won't work much.
Hmm. PVA is uh, PVA
It's
like
a synthetic
I'm not
one. No,
sure. Yeah, synthetic one. I'm not sure about Merrill whether they are manufacturing themselves or not.
no, they
But
are not
here
manufacturing. I
they are
know A to Z of
manufacturing.
Merrill. Like, I have all of
Okay.
my colleagues in Merrill.
Acha, I'm not sure about that, sir, because I haven't got exposure to that department when I was in Merrill as a consultant. I was mostly into endoscopic part for them.
Yeah, because I was there with Merrill for some time, where we worked on biodegradable stunt, uh, myself and one other guy. Then I moved to Merrill Clinical Division. That's there in Ahmedabad. That's Clinex. Dr. Ashok Thakkar was there. So I was training them on all this clinical writing, all the stuff. I was not doing much, but I'm reviewing them before publication. I mean, before we submit for publication. Okay.
Oh,
Yeah, I know. I mean, I'm more acquainted with companies in West than in the South. South, I know like 5, 10 companies, but Maharashtra, Gujarat, I know all companies. I know all
that's
SMEs,
great stuff.
yeah, big companies because I worked there for a long time and I like Ahmedabad. Yeah, guys, like I said, we'll have a good session today and And in between, we'll have a break. If you want to have snacks, or you can, if you want to have directly lunch in the break, have lunch and keep listening to me. Because I'm going to take a very important topic. I told you this topic on risk management. Okay, is like, you can assess this like it's a brain or central nervous system of the QMS and the devices, or it's a heart. To me, it looks more relevant like the nervous system because it connects every part of the body. Okay, and it actually does a lot of functions of not only connecting, but blood also connects to a lot of part, but nervous system connects and does a lot of activities, whether it's voluntary, involuntary, systemic. So that we'll be discussing today. And I know like it's very, very important. Uh, Risk management is a key, it's a basic for everything. And I know like somebody who is good in risk management, okay, uh, I know he's going to have a great future. Because once you, to understand risk management, you should also have now knowledge of couple of other things. It's not like you understand risk management and do everything. You need to have an understanding on 13485 how it is linked. You should have understanding on the clinicals. You should have understanding on PMS. You should have understanding on usability engineering. And most importantly, you should have understanding on design and development. So these 5 areas are the one where PMS is, sorry, uh, risk management is so closely linked. Uh, So it's important for everyone, every division, every department. Okay. Maybe in some companies I've seen guys who are specially recruited for risk management. And I know like companies where the departments itself, particular department work on risk management, like design and development do their risk management. Uh, like the PMS, they do the risk management complaint. It's totally integrated. Like so, whatever may be the case, you may have an individual looking at the risk management, whether you have the individual department working on risk management, this is going to be key. So, risk management, I am going to discuss for 9 hours. That's almost like Saturdays on 3 sessions. Okay. It's like we are going in depth and we'll have a very good practical approach of, I'll give you some work. Okay. I'll go in depth in details. I'll also give, not lengthy work, like something like on assignment, a simple thing like how you grade the severity, how you grade the occurrence based on your own company's medical devices. Because I know like where Aman is working, where Pranitha, okay, Pranitha, I know. Masjid, yeah. I know Masjid was working on hemostat, but I forgot. Where is Vaibhav? Yeah, Vaibhav is there. I know Vaibhav is what he's doing. And Madhu and Soman, their company is so close to it. So close in the sense I'm not their employee. I studied their company, okay, from head to tail. Uh, it's not that I'm joining their company, but I was interested in the company because they are in a very advanced medical devices like on surgical robotics. All this stuff makes me interested. So, just to make me understand, like, uh, did I discuss with you last session integration, those who are present, integration of ISO, integration of USFDA 21 CFR with ISO 13485?
Yes, sir.
Yes?
Yes, sir.
Yeah. So I'll give you 5, 6 MCQs. You attend that. Okay. Answer them in chatbot. In chat, not chatbot. I'm getting used to chatbot because I use it. Because I mean, I'm sorry for asking this question, though I discussed like uh, I'm traveling a lot. I've become like a vagabond. I'm discussing a lot of things with a lot of people, and I forget sometimes like to whom what I have discussed past. Okay, and of course I have a short memory, not like Gajini, but I have a very short memory. But those short memory, I have a very good logic and reason. So share that. Try to attempt it. I mean, it's good like when you entangle or integrate and modify the ISO with 21 CFR. I mean, I feel it really great. Uh, I told that why this happened, why not earlier, why history made this. This is going to be a big history in the medical devices QARA, where US is coming, but it's not a joke. Like you need to take seriously because I know how the US FDA audits go on. Okay, though they come under the ambit of ISO 13485, they will be having, okay, they go evidence based, in depth, effectiveness. They look at all superlative degrees, superlative words. Okay, they want everything to be the best, not good, better. They want best. Okay, and they have enforcement of this. They enforce it. It's not like you just need to. It's not voluntary or something like that. They enforce that you need to do. Okay, a lot of things are there. Though I discussed, I think I also shared this document with you. You have not shared that. Today I'll do it. Uh, I request companies like maybe Intuitive doesn't need because they already have MDSAP. Maybe Aman, Majid, and Vaibhav can work on implementing this at early stage. It's not very difficult. Okay. And when I say not difficult, you need to do some, I mean, groundwork, spade work on that. And I mean, of course, it depends upon the management whether they want to adopt it or not, because Aman said that he's going for 510K. Yesterday, there was some long discussion on WhatsApp. So you can adopt it very quickly. And I have all the QSPs, sorry, SOPs required for that. Okay, if you want to implement it, talk to your management. I'll share you, I'll help you in implementing this. Even, I mean, it's like implementing ENISO. Now we have, that's for Europe. QMSR is for US. Okay, like how we have done for ISO 13485. It's like doing QMSR, Quality Management System Regulation for US FDA. But I know like it will be a bit tough. Documentation won't be tough. Okay, implementation and auditing, they won't call it as audit, they call it as inspections, okay, or they call it as a investigation. Okay, they don't call them as auditors. So that will be tough. Because I mean, I know like, you know pretty well, US always try to benchmark their standards and Europe follows the same. So I mean, I'll give you 5-10 minutes, just go through this. I mean, it's for two things. For me to know, I mean, for me to know how you understood this, uh, and whether I need to view some capstone project on this, because I'm planning capstone projects for all new updates that have happened. Okay, one update today I'll be covering. One that happened is on this integration. There are 3-4 updates that happened since we started on November 7th, our sessions. Okay, updates on UMDR. This is a very crucial one. Okay, and mainly the back compatibility, which I'll give you an overview of that. Okay, that happened in 2025. I also shared like 9001. There were some changes. New version has come in 9001. That is a very generic quality management system. Okay, it's like any company can have 9001, ISO 9001. But there were some changes that happened in 9001. though it is not important, but as I said, the origins of ISO 13485 are from 9001 only. Okay, so some of them we need to adopt, and don't be surprised if there is a new edition that comes in 2026 on ISO 13485. Most probably, I think it may come because there are some working groups called, ISO working groups, all from 140. In ISO, you have like 146 countries. Okay, you who frame this all ISOs, IEC, all this stuff. They are working on this. Okay, whether to come out with the new edition in 2026 or just stay with the amendment. Because of two things, I feel that they may come up with one, new edition. Because one is it has become QMSR 21 CFR 820 has come in. Okay, in ISO 20, with 13485. And the second thing, what I feel is like there are too many amendments that have happened since last 10 years. Okay, if you look from 2016, so that amendments has to be imbibed and regularized. They should not remain as amendments. So that's second thing. And third thing is like after every 10 years, it's not mandatory. After every 10 years or a decade, you'll have the revisions of these 13485 standards. Of course, one time this missed, like in 2003, there was a new version released, new edition released, but it didn't happen in 2013. It happened in 2016. So there was delay for 3 years. Maybe I don't know, like it may be delayed or delivered. The other is like, that's what 9001 also says. The new 2025 or 26 version says that they are integrating more of the software linked devices. Okay, adoption of AI. I'm talking about 9001, ISO 9001. Okay, and in medical devices also I discussed a lot. Like in future, you will see most of the devices linked with IoT or ML. Okay, it's for better monitoring, better healthcare system, and mainly for reaching to the rural areas, and mainly addressing the elderly persons. Because I said after 2050, India will have the most aged population. Now we are good on the demographics that we have. Perhaps we are the one country which has the youngest population with an average age of 29 years. But this will go down. After 15, 20 years, India will become like China. Okay, China or Japan. Like they have they have, they have most elderly population who can't work. So the workforce there in China and Japan is very less compared to India. We have good workforce, but we need to adopt the technologies. So, these are some of the reasons I said. But if you just attempt, take 5 minutes or 10 minutes, how much time? Just to have an overview of like whether you understood this or can you adopt it. Okay. Because why I'm talking is I know like a lot of people are, going through our recordings, okay, because of so many issues. Because when I see the analytics of the recordings, I see, I don't know whether it's our group seeing more or other people seeing more. I could see like there are a lot of people seeing our recordings. But I told like maybe after this session, I will share recordings personally to you, not in the group. Okay, like I was doing for AML classes, so that and I'll make it private, not in public domain. So what we'll do is just go through these guys. Now here you have, if you If you're not able to get the right answer, also it's fine. Just to see how you understood this, or can I give a capstone project on how you can integrate this so you can see on the screen? Because there's something like 8 MCQs. I know that some of them are a bit tricky and logical. Some are basic. If you're done, then we'll go ahead. Yeah, good. Most of you answered right. Yeah, most of the answers are either B or C. Okay, done. I'll give you the key at the end of the session. So I mean, like, next half an hour, what I'll do is up to 7:30. I'll discuss something on 10993. What are the changes that happened compared to 2018? Okay, because this is quite important for all the organizations who already have done the biocompatibility studies for their devices, and it's most important for those who want to do biocompatibility studies, because I said ISO 10993 2025 document is already released in December, and there is a lot of emphasis or enforcement from the auditors, at least to change your biological evaluation plan, okay, and do the gap analysis and report to us like what are the tests that are pending from your side, and give timelines like when you are going to complete. For those who already have the test done, biocompatibility, biological evaluation test done, the notified bodies are asking, even CDSCO is asking, okay. FDA has, I mean, I'll talk on that. FDA has a bit different in terms of their biocompatibility studies. 90% is the same, but they go very in depth, like on say though teratogenicity, I mean, cause you if you do carcinogenicity, okay, like they still provoke you to do the teratogenicity studies, because if there is no carcinogenicity, I don't think it will cause teratogenicity. Okay. Uh, I mean that you can give a justification, but still they ask. So what I'll do is, because it's quite important, one topic which we discussed is on integration of 21 CFR 820 with 13485. The second important thing, which I may not take much time, because I want to get on after the break, I want to get on with 14971. Okay, let me make you understand. Uh, there are some 7-8 changes that happen, but they are very significant. Okay. Yeah, first let me hear from you, like how many of you, I mean, understand biocompatibility studies? Like you're new to it or you already know something on biocompatibility? It,
Sure, it is a study on the material which is used on in device which is come in contact with the patient.
yeah, that.
Is
Yeah, I know, like,
biosystem.
yeah, basically we study about devices which are in contact. Okay, it could be short or prolonged contact or long-term contact. Like if I look at your devices, there could be a short term. I'll tell you like why your device also has to make some changes in your BEP. Okay, because of the extractables and leachables, and if the device is reused, okay, the duration also changes. The particulate matter is not considered earlier, but now the particulate matter is also a key one. I'll tell you all this stuff. The 7-8 changes. I'll if you require, I'll share with you later the new biological evaluation plan (BEP). Okay, and how to integrate this. Here I'll touch upon the gap analysis and all that part. Can you see my screen? Okay, this is 2025 edition. You know, like it came in December last year, and it was open now in domain, and every organization need to adopt this. It's no more in a stage of transition, something like that. It has to be adopted because the guidelines or the standard has come out, and you need to at least, as I said, no, show some signs or give signals that we are adopting this. So I'll tell you stepwise like how you are going to adopt. But before adopting this, you should know what are the changes. Okay, what are the gaps? For somebody who is going for testing of their new devices, they should know everything. But those already who have, got their back compatibility test done, they should close these gaps. Okay, if they don't close these gaps by their next audits, okay, I think you'll get major NCs or some red flags. Okay. So what it sells is the biological evaluation is firmly integrated with, I know like earlier also, okay, it is integrated with risk management, biological evaluation, but this time they made it like what you call biological evaluation risk has to be a separate document. Okay, the risks that are coming from biological evaluation, like how we write the risk management report. Okay, like we identify the hazard situation, harm, risk evaluation, analysis, risk controls, all this stuff, we give it in a tabular form. Okay, like that, even for biocompatibility, you need to provide it separately in your biological evaluation report. Okay, you need to provide separately what tests you are doing. Okay, and what is the basic biological endpoints and what are the risks they are carrying. I'll tell you in depth risks in the sense if you have not done the test. Okay, if though you have done the test, still there will be some residual risk in them. Okay, that you need to give a justification. So it is totally embedded. You need to have a complete logical sequence of how you are doing it. Okay, and give it in a very structured flow diagram way of how you are doing the uh, biocompatibility studies, and in parallel looking at the risk management. Okay, and this is, since risk management applies throughout the life cycle of the device, your biocompatibility studies also has to be linked to the risk management throughout the life cycle of the device, because in post-marketing surveillance, you may find some, no, some adverse events are not so adverse, but some events or some complications that may link to your biological evaluations. Okay, I can tell you a lot of things on basically on sensitivity, chronic toxicity. Okay, you may stop your studies up to acute or subacute toxicity, but if it is a chronic toxicity, how you will justify, though it is for prolonged between 24 to 30 days, but if it is causing chronic toxicity, okay, that's where you need to link up, and the expectations on material characterization, exposure time, and traceable justification. This is very important, like as Madhu told. We need to do a material characterization. What is the exposure assessment? Earlier, we used to say that the exposure of the device is for as a limited time, like up to 24 hours, or there were three categories, like up to 24 hours, 24 to 30 days. Okay, that the other is permanent implants, like more than 30 days. But now the total exposure based on 2025, you need to relook into what is your exposure time based on the reuse of your device. Okay, suppose if you are reusing the device, it could be off label or on label. If the device is reused, okay, the exposure time will amplify. Suppose you used it for 6 hours in one patient, and the same device you have used it in another 5 patients. Okay, so it is from in between 0 to 24 hours where it is in limited contact. Now it is enters into a prolonged contact because you have reused, and there will be multiple additions to that, or you need to multiply it into the number of hours you use. So a lot of devices from limited will enter into prolonged. Okay, and some devices in prolonged contact time will enter into, okay, permanent contact. So that you need to be careful. You need to assess like how much is the contact time for the primary use and reuse, and multiply that and see like what is the total contact time. Okay, now you need to take total contact time, not just one time contact time for other device. Suppose if you look at the devices that are used in, I know like lot of devices are reused off label. If you are using off label also, still you need to consider that we say it's single use sterile, but I know like auditors know like it will be used multiple times, and mainly there are some devices which we say that can be used multiple times after re-sterilization. Okay, I know a list of devices, you can check it out, list of devices that can be reused multiple times after re-sterilization if it's a single use also. Okay, and there will be some, like I can take example of in Q2 where in the robotic surgery the material will be in contact with the human organs. Okay, or where the U device for stitching or something like that, or at the high-end robotics they look at, it will be in contact with patient. Okay, not one time, multiple times. So it is in multiple times. What is the contact of the device or contact of that particular material? Okay, suppose they have done 10 surgeries. So they need to cumulatively tell what is the contact time in 10 surgeries. Okay, then it will either go to, I mean, it will change, there will be some change in the contact time. Okay, whether it's limited to prolonged or prolonged to permanent. I think it may not go to permanent, maybe it'll stay in prolonged. And the important thing is like whether those that are used, basically alloys or metals used in robotics or others surgeries, they have a lot of, because they are alloys, there is some issue on leachables or extractables when it comes in contact with blood. So that study has to be perfectly explained. Okay, and in that what we call toxicological risk assessment. Okay, if it enters more than the permissible limits, what is the assessment you need to do? Okay, I mean, I'm just quoting example of that. There are other lot of things which need to work on. Okay, I just said a few. Okay, there is emphasis on particulate matter. Okay, contact time counting. Now, I mean, we never discussed on particulate matter for a lot of devices. Now, for every device, you need to do, I mean, explain what is the particulate matter. You know the, it's not only the number, what is the size of the particulate matter. Any device, okay, any device in contact with human body, you need to write, you need to add one more specification in your uh, I mean technical specification sheet or in your COA, that the total particulate matter is so much on the device and what is the size of it. You may have from say 10 nanometers to 100 mm, because this when contact, basically you can look this where it is in contact with the systemic circulation of blood contacting devices, devices that are in contact with blood. It causes serious concern, mainly in formation of clots or embolism and all this stuff. So this is also made strict. But let's categorically look into one by one and see what best we can do with all these changes. Okay. I'm just picking up the 7-8 major changes which will have a lot of impact, and you need to change your biological evaluation plan as per these changes. Okay. Risk management integration, biological evaluation becomes a part of risk management life cycle. I mean, this is not new for us, but every time we used to link risk management with, traditionally we used to link risk management with either clinical evaluation. Okay, off late we are linking it with PMS. Okay, off late I know, like earlier it was not even linked to PMS. Because PMS gives an inputs, that's the important criteria. PMS will give you lot of inputs in the risk management, whether your complaints or your risk events, COPPA, all that has to be linked, imbibed into the risk management. Whether you have new risks or the old risks are repeating, you need to have more risk controls, effectiveness of the risk controls. Okay, here in biological evaluation also, you need to, as I said, you need to have a separate section to write. Okay, I'll share you the BEP. In your biological evaluation plan, you need to write that, like, what is your decision making tree or flow for biocompatibility studies. Okay, for everything you need to give a justification. Why you have done this is fine, you can give justification. And why you have omitted others also, you need to give a justification, which was not earlier there. And when I come to the risk management, it is pretty clear. Okay, you need to have this, like how we do in risk management. In biological evaluation also, you need to identify the hazard. Sorry, know what is the biological hazard. Okay, hazardous situation, because you know pretty well all hazards won't become harm. If the situation, I mean, if the environment or the condition has a hazardous situation, then only the hazards will become harm or risk. So you need to know, I mean, that's why I'm saying you need to write a separate table, like what are the hazards. Okay, you may do cytotoxicity. Why you are doing cytotoxicity? Okay, there could be some, when it is contact with these cells, it may cause some cell damage or damage to membrane, cell membrane. If you look at the cytotoxic studies, you know, like basically they are in vitro, okay, done on fibroblasts. Is there any change in the cell morphology, cell structure, cell functionality? Okay, I mean, that is hazard. What is a hazardous situation? Okay, where the situation makes this hazard convert into an harm or risk. Okay, like suppose I say I have used this material in an compromised, okay, area where there could be a lot of bleeding or there could be a lot of burns, injuries. Okay, obviously this material what you are using will affect these cells that are used. It could be any cells in the human body, any organs or tissues or cells. Okay, that hazard could turn into a hazardous in that particular situation. Okay, this hazard may turn into an harm. Harm is nothing but the risk. Okay, in that condition, what are your, how you are doing your risk analysis? How you are evaluating? What is the risk in it? Suppose I am using a material, there is a hazardous situation. It is causing harm like inflammation, or it may be causing like acute to chronic inflammation or bleeding, or it may aggravate the burns, or it may cause too much of blood leakage. You can write so many things. Or cytotoxicity itself, Okay, in particular hazardous situation, it may form clots if it is not compatible with blood components. Okay, of course, for blood components, there is separate section on checking the compatibility with blood and blood components. But these are the harms that may have. So what are your risk controls? Okay, and what is the effectiveness of risk controls? So all this, I'm just telling you example, like you need to describe all this, like how we generally used to do in the risk management files. Okay, and you need to write this. Of course, in risk management, we used to have a section on biological evaluation. Okay, but it is very short, like whatever test you say, you do the test, give the test number and say this is the standard, and at end you'll write it is non-cytotoxic, it is a non-irritant, it doesn't have any skin irritation, skin sensitization, it doesn't have chronic toxicity or subchronic, all this stuff. It doesn't interact with blood and all this we used to write. But here you need to write them in separately in biological evaluation report, starting from what is the hazard situation, how you that hazard may convert into an harm, and what are your risk controls, effectiveness of controls. Basically, you need to do a lot of risk evaluation. And here I am telling you, I mean, when I come to 14971, I'll tell you there is nothing as risk is acceptable. Okay, I'll tell you there is difference between 14971 and what EUMDR says as per the new annexure, that is Z annexure that came in 2021. They won't agree with this, that risk is acceptable. All this they won't say. I mean, you need to define risk, or you can't say acceptability of risk. Okay, risk is always risk. Okay, all this you need to write it. What is the estimate? We discussed like risk index. Risk is severity into probability. Risk controls, documented residual risk. So this is now, earlier also it was there, but it is now we need to give a lot of explanation on that. Okay, and the justification of why the residual risk does not affect, okay, in compromised situations, the residual risk doesn't have any issues on the safety. This is the main thing which we need to understand. You need to give justification of your residual risk. Okay. Though your residual risk may come into the green zone in the mapping, but still if you have residual risk, what will be the, I mean, the worst case scenario that your residual risk may affect? Because they are going in depth on this. So you need to have a separate risk management report for biological evaluation. I think that sentence tells you the importance of biological evaluation, what changes that have happened. So you need to incorporate separate risk management for biological evaluation in this form, from identifying hazards to risk controls and residual risk. Okay, that's what I said. You need to change your BEP, BER must be changed, and it must be traceable to the risk management file. This is very critical. Whatever you have done in BEP, BER, whatever comes to your biological evaluation report, you need to link that to risk management, because you write risk management separately, risk management report, biological evaluation report separately. So the best thing is you give the reference number of it and link it to the risk management file where you write it separately. So you should find an integration of the biological evaluation with the risk management. Okay. Of course, if you are doing for Europe, that is another big thing, that you need to link it back to GSPR.
So
So
I
no,
have a doubt over here.
yeah.
So what if the residual risk is related to user, like the risk which cannot be controlled from the manufacturing side? So in that case, what should the manufacturer do in such cases?
See, that's why if you are not able to address the residual risks, and you should provide what are the risk controls you can have, risk mitigation. You should have some controls over that. See, Majid, it's not like you can have zero risk for the any device. Even simple needle or cotton will have a lot of risk. But what are the residual risks? We'll discuss on, I mean, when I come to risk management, I'll tell you what are your risk controls and effectiveness of the risk controls. Okay. It's not just writing that these tests are these documents or these records, okay, this IFU or some training, these are my risk controls. I'll tell you on IFU also. IFU is the last option for risk controls. Okay, mainly risk inherited by design is taken. Okay, that should be the first option for risk control. Second is the procedural aspects. Okay, when you are doing the procedure using the device, what are the risks? That's how you are controlling the risk. That is the second option. The third is on the training, IFU, other documents should be the third choice. But preferentially, if you look at risk by design is very important. Then risk that you can control, okay, during the process of the procedure is important. Whatever process, your internal process, sterilization or production, XYZ, okay, on your bioburden and all this stuff. And the last option is, I mean, this is the last option. And there is also a conflict of information, or there is a big debate still going on. Of course, it is not settled that you can't use some of the training or information that you have passed on to the end user, like the IFU training material, brochures, that as risk controls. Your IFU will not become a risk control, though it says that it's the last option. But EUMDR, that is that's a point with Europe. Like we have ENIS4, okay, for QMS. In risk management, they have come with EN 14971, ENIS4 14971. They have a separate risk management. I mean, separate standard, okay, linked with the regulations on risk management. It is called EN ISO 14971, like how we have, okay, in the QMS, EN ISO 13485. Okay, we'll see whether when I discuss on risk management, I'll tell you. But of course, there will be residual risks. But the residual risks, what are your risk controls? If you don't have risk controls on the residual risk, either you want to change the design. I'll tell you one important point. If there are risks, residual risks that have high impact on safety, okay, then there is no choice, but you need to, I mean, bring it to what we call as low as possible. There are two words which will be used in risk management. As earlier, we used to say as low as reasonably possible. It is called LR. As low as reasonably possible. Now it is like as low as possible, because they have taken out all the economic considerations. Say, if I say that if I want to bring down the risk as low as possible, I need to do a lot of design changes, that it will cost me so much of money. I need to do again some trials or clinical validations, something. At least a part of that where you are addressing the risk, that you need to redo it. So it costs some money. So what they have done is like they removed the economic consideration. How much may it cost? If that risk can be reduced to as low as possible, you should remove it. Okay. After removing whatever, you should have some residual risk. That is why in risk management, design by risk or risk in design is very, very important. And the other thing is like when you are doing, I mean, working on the residual risk, there is a this is a very crucial point people in design and development understand. There will be some parameters or some design inputs. Okay, if you reduce the safety, it may directly correlate or link with the performance also. Suppose if you are reducing the safety, the performance also comes down. Are you able to understand my point? I know, like when you work in design and development, or when you take some of the design input, I can bring down residual risk as low as possible, but my performance is also decreasing because this parameter is interlinked with both of them. Suppose think of a material which is doing very great in performance, okay, on expandability or radial strength, this, but it is having some safety issues on extractables or leachables, mainly. Like you see, nickel is the one which leaches much faster than most of the other alloys or other metals. Okay, nickel toxicity is very dangerous. It may cause, prolonged nickel toxicity causes cancer. So, but for me, nickel in that alloy is important because it is contributing a lot to the performance. So, how should I balance it? Should I bring down the nickel content in that particular alloy? My performance comes down because if I bring down nickel, there may be a recoil of that material, or the what we call longitudinal compression will be there. It won't be so elastic. So in that cases, okay, what they say is you can bring down the risk, okay, as low as possible. Still, you can compromise on performance. Okay, this for very high risk scenarios. If the risk is very high, okay, in that case, you can compromise on your performance, because this risk and performance, or what we call benefit and risk, that analysis will do. What is the benefit risk? That time you should compromise on your benefits than on the risk. So, a lot of things are there which we'll discuss. That's why I told you that this is a very interesting topic. I like the most. It's not like because I like the most, I'm explaining it for 9 hours. It's important in QMS or in your devices also. Okay. See, okay, this is, as I said, it has to be totally integrated with risk management. But why I'm telling this much before talking on risk management is, you can link up this when I'm talking on risk management 14971, how it is linked, biological evaluation as a risk management, okay, can be linked. So it's BEP is not, biological evaluation plan or report is not a standalone document. Okay, I know, like in all of this technical documentation, what we do, I don't find any single document as a standalone document. It will be interconnected because the processes are connected. Okay, no document could be standalone except one document which I see is the GSPR. I say it's standalone because it connected to all the documents. So it can be a standalone document, that is the GSPR Annexure 1 in UMDR. Okay. It is no more a standalone in BRC that merely lists tests. Okay, each test decision must be linked to biological hazard risk estimate. So each test what you perform has to be linked with the hazard and estimate the risk. I'll tell you all this stuff, how to estimate the risk, like how to look at the probability, because this is a very important question. When you come to the risk manager, how you calculate the probability. Okay. Probability is graded. Okay, when you come to the quantitative or even the semi-quantitative analysis in the risk management, this has to be quantified or graded. How you do that? Okay, I'll tell you like how you do it with hazard situation is called P1 and P2. P1 into P2 gives you P occurrence. Okay, then what about severity? Okay, how you calculate severity? Because when you write a risk management file, I know like lot of the things when you give the risk probability or occurrence and some of the uh, risk analysis, what you do, okay, most of the things are very subjective. Okay, you may think that this is going to be having high occurrence or high severity. I may think in a different way. I'll tell you also on that what is the knowledge necessary to write a risk management file or in assessing the risk. Okay. So I mean this, remember it very blindly. It is biological evaluation or BER or BEP is not allowed, is not now a standalone document. You need to totally integrate it vertically or horizontally. Okay, with biological evaluation, sorry, with risk management and risk management in terms of the product life cycle. Okay, when I say product life cycle includes from design till the PMS. Okay, this is what I said. You need to find the hazard situation, endpoint test, results, residual risk. I'll share that BEP plan, I mean BEP, biological evaluation. And second, as I discussed a little bit on that, reclassification of the contact time. So reclassification of the contact time is not for a single use. Okay, suppose my contact time of the device is for say 6 hours or 3 hours, and if I reuse that again, okay, maybe on label it could be there. IFU message that you can use it, because that's the reason I said most of the devices are reused. Some are reused off label, though we say them it's for single use. In that situation, I said it will be, if you use it for 5 times, it will become a cumulative contact time of the 5 times you have used. That's the reason in the category of contact time in ISO 10993, you have 3 categories. Okay, limited, prolonged, and permanent. Limited is 0 to 4. Prolonged is 24 hours to 30 days. And permanent is more than 30 days. So here, I mean, you need to look into your device and come out with an answer. What is your cumulative contact time, not one-time contact? Okay, definition from contact duration of device categories were revised. Manufacturers must justify how they count contact days or contact time, including, this is very important one, foreseeable reuse. Okay, we know that, or misuse. You need to take into account, okay, what is the foreseeable reuse of it. Okay, they may resterilize use or the device itself. I know like a lot of catheters or like a lot of equipment like needles, they resterilize, or drip sets, they do a re-sterilization. It is there in the IFU, like you can resterilize and use. Okay, it's a foreseeable reuse of it. But the other is misuse. Okay, if it is totally misused, because your IFU is not telling them to reuse, then it will become a misuse. But again, I think I told to someone, still it is your responsibility only. Why this is important contact time is your, I mean, once where it is a limited contact time within 24 hours, move to prolong. Some prolong may move to, I think there is very less chance prolong may move to, I mean, permanent. But there is a very thin gap between this. Like if it is 30 days, if I say it is 32 days, then I have to go for permanent implant. All that, I mean, there will be a lot of tests, like it is going into high risk category, and then you'll have a battery of tests. I mean, that we need to look into a clear explanation. And the reason why they say contact time is very important, cumulative contact time. Okay, one is basically because of the extractables and leachables that happens. Like this I have seen in dialysis catheters or urinary catheters. Okay, when the contact time is more, okay, and some of the dialytical catheters are discarded or they re-sterilized and used. Okay, the extractables and leachables from that polymer. Okay, there will be some polymers which are so dangerous. Okay, I talked about nickel, no? In polymers also you have so much of polymers which I mean in the circulation, in the blood circulation, they may leach out or they may extract from it. Okay. And if it is a prolonged one, they may cause a lot of, I'll tell you one, they may cause a lot of neurotoxicity also. Okay. And that's why some tests like neurotoxicity now became mandatory, where we never tested devices for neurotoxicity. We never tested devices. To my knowledge, there was no test on neurotoxicity. There's no test on immunological toxicity. But all this now came in that you need to do this neurotoxicity also. Okay, but some part of that was covered in chronic toxicity studies or repeated dose toxicity studies. But now they are saying that you need to exclusively do it for neurotoxicity or immunological toxicity. Okay, and why this matters, the cumulative contact time. This is what I said. Devices previously treated as prolonged may be reclassified as long term. This is a very high risky statement. Like prolonged, when it is moved to long term or permanent, like you will have to spend a lot of money, because for long term or permanent, they will have almost like, to my memory, there are almost like 15 tests. Okay, and some of the tests are very, very costly. Like I'm talking on carcinogenicity, teratogenicity. Okay, but all of them are because of gene mutation. I don't know why they asked to do all this stuff. Uh, Chronic toxicity, repeated dose stuff. Chronic dose is a 90-day study. Okay, acute, subacute, they have to done in animal models. There is no way that you can do in vitro. Okay, like I mean, as we go down, the cost keeps increasing. I mean, this is prolonged to long term. Under the new counting rules, triggered additional endpoints. Okay, this is what I'm saying. Like on chronic toxicity consideration, additional endpoints is maybe if your device is up to only 30 days contact, it may be limited to acute or subacute, somewhere in between this. But when it enters into a long-term contact or permanent contact, it moves from subacute to acute into chronic toxicity. You need to do chronic toxicity there. That too in higher animal models like in rabbits or in rodents like rats. Okay, and it will cost a lot. Okay, and foreseeable misuse, reuse, use longer than intended must be evaluated and map up, classify, exposure. Whether is strong expectations of material and chemical characterizations is already I spoke. Like the first step now in any medical device is that you need to do your material characterization. Okay, material or chemical characterization. It could be metals, polymers, or some organic compounds or inorganic, plant derived, human derived, animal derived. First, you need to do the chemical characterization, okay, and material characterization of the devices. And you should know, like, generally we do it in semidex for metals, even in for some of the polymers also you can do semidex, or TEM also you can do. Scanning electron microscopy with EDX. It will give you the composition. I mean, you need to quantify it. So Semidex is used for only qualitative purpose. It will tell you what are the metals or polymers that are present. Okay, but it can't quantify. So you need to adopt methods like analytical methods, like whether it's GC gas chromatography with mass spectroscopy or LCMS, HPLC with MS. Okay, ICP, it's also linked to ionization cryptographic protocol. So what I'm trying to make you understand is, before you start your biocompatibility studies, the first step is do the material characterization. First, you know what are the materials or what are the elements, whatever you call metals, alloys present in your finished device, okay, in your finished product, not in the raw materials or in the intermediates. Okay, because there could be some, I mean, the reason why I know they have stressed this is, if you test the raw material, there may be only 5, 6 elements, but in the processing stage, okay, in the processing stage or in the manufacturing or in the production stage, what happens is these polymers or the metals or some may deform into new metals or new polymers, some may degrade because of the temperatures and the humidity and a lot of things other use. Some may form, I mean, because of due to some chemical reactions, then some may get oxidized. And at end of the day, your device will have, okay, some new chemical entities or new metals that are not seen in the raw material because of the processing. These new materials or the new ones which have come out because of the processing may pose toxicity. So for your finished material, finished product device, you need to identify what are those elements or polymers or the metals that are there. Okay. Uh, it could be from, I mean, the raw material could be derived from any source, and quantify them. Quantify them through various analytical methods. Then once you quantify, you know, like uh, whether they are within the controllable limits, what we call it as LD50 by ED50. I mean, your material data safety sheet, MDSS, shows that, okay, what is the allowable quantity in humans. Mostly it will give in animals, but you can interpolate it to humans out of what is the allowable quantity of this particular thing in humans. If it is okay, fine, or if it is crossing that limit, when you're quantifying it, then you need to do the studies. Okay, because the new things, new elements may cause new risks, or if there are leachables or that are leaching out of them, it may cause risk. But I'm bit pragmatic on this, like it happens in very sensitive cases or mostly in vital organs. You see this. Mainly you'll see this in implants. Okay, maybe in class B, class 2B devices. So early analytical chemistry, like using all this analytical, is required to identify potential hazards and set priorities for biological testing. And after that, you need to do the toxicological risk assessment. This is very, very important. Like what we call LD50 by ED50, generally we call it as a therapeutic dose, but more here we need to look at LD50. Okay, ED50 is the effective dose, but LD50 is the lethal dose, which kills 50% of the cells or 50%, whatever it is. So what is my LD50 and what are the concentrations in this? This you can reflect back into cytotoxicity and see how much is the cell death. Okay, you need to interconnect a lot of studies within the biocompatibility to understand toxicological risk assessment. And toxicological risk assessment should be one of the subject or one of the topic that has to be discussed in your biological evaluation report. Okay, maybe when you write the report, let me know. I'll tell you like how to write this toxicological assessment report. Okay, it's not that I'm expert on it, because my basic post graduation is on pharmacology and toxicology. Though I've done a lot of things, but this keeps me interested. and rely on these data sets. So major change 3 is you need to do chemical, material and chemical characterization. This I told you, particulate matter, this is something new. You need to have a new SOP, or it has to be included in your BEP, because you need to tell for anything that comes in contact with skin, with contact with human body. Expand expectations around particulate release, whether they are getting released, the particulate matter, it could be, released or get added to the device. Okay, particle count, size distribution, morphology, and biological relevance must be evaluated where relevant. See, this is very, very tough job to do a particle count on any devices, basically for those which come in contact with human body and more importantly with the blood. Okay, because if you look at the 1493 flowchart, there will be three things. Like one is contact time. Okay, duration of contact, as we discussed, there will be three categories. The other category where we need to link up that matrix of table is in which area it is going to get in contact. It's going to be on the surface, or it is not at all contacting, or just on the skin surface, or a breached or compromised area on the skin, or it is getting contact with the circulating blood or not with the circulating blood. Okay, or it is for intravascular surgeries where it is totally connected with the central, circulating blood with the heart. Okay, so that is one category, and the other category is on the duration. One is the contact contact area where it is going to get contact. The other is the contact duration. So you need to map up where it is getting contact, what is the duration. Then you need to look into a lot of other things like I explained. What are your basic biocompatibility studies in reference to the new document? Okay. Cumulative exposure time, what are the leachables, extractable, is it linked to risk management, how it is risk linked, whether you have been interconnected. You need to change your BEP and BER. Okay. Because here I said particulate matter, particulate analysis, data interpretation, source distribution. You need to need a source also where from the particulate matter came. Attribution, worst case scenarios. You need to do this particulate risk assessment, especially for surface shedding or indirectly contacting it could be indirectly contacting, but it may shed a lot of particles, because this could be a risk of acid. Now, when the particulates form aggregates, okay, it could be very small, but when they form aggregates and enter into circulatory system, it's very dangerous. Okay, like they enter into the circulatory system and they go into the lungs. If you have pulmonary embolism or go into the heart, there may could be chance of clots or platelet aggregation that causes because any foreign body reacts, there could be a lot of immunological responses. So I think that could be the reason they made, but I don't know why earlier it was not focused. Okay, expanded biological endpoints and conditional testing. See, this is what I said. Now your endpoints will expand on lot of things: contact time, duration, particulate matter, okay, risk management, and totally you need to restructure into your 109931 flow diagram. Okay, duration of contact, in which tissue it is contacting, and they have also added something like immunotoxicity, neurotoxicity. But these are very difficult, like only in behavioral studies you'll know neurotoxicity, that too in animal model. Very difficult to do in vitro. It can be done in vitro on certain neuronal cell lines, like on rat neuronal cell lines. We can know whether they are causing any neurotoxicity or degradation of myelin sheath. Because Majid can remember, like any remnants of the hemostats, generally hemostats are the one which absorb blood. They have a lot of absorbency. Okay, some degrade, some may not degrade, but substances like gelatin won't degrade, ORC may degrade. But if there is a remnant of that, okay, in very sensitive neurological nerves, like it can be left in optical nerves, or it could be left in nerves that are very important, like in the cardiac nerves. Okay, if that is left, there is a high risk that it won't degrade. Okay, and it will interfere with the nerves, and it may cause, say, I know, like what are the problems with hemostats? They may cause paralysis. There are different words for that. But the worst condition is they may cause paralysis, or if it affects the optical nerves, you may lose sight. Okay, or there will be degradation on the side. So this immunotoxicity may happen because of the material itself degrading or the leachables, or because of the particulate matter, and there is neurotoxicity. So these are new endpoints which you need to look in. And yeah, it's not like I don't recommend doing all this stuff, but you should understand that maybe in the material characterization, material and chemical characterization, you should be able to understand whether the material has any effect on neurotoxicity and immunogenicity, and give a better justification. Okay, instead of doing all these studies, I know it adds a lot of lags. If for new devices, if you take class 3 implant with these standards, with 2025, I think the budget may cross 1 crore. Of course, we do it once in a lifetime of the device, but still it's too much. So it broadens list of biological affiliates to immunogenicity, neurotoxicity, but this should have been covered in chronic toxicity studies. Okay, toxicologists must have conditional decisions based on chemistry exposure. Okay, the major change is this, or I don't think this is a big thing. Competent evaluator and documentation requirements. See, you need to have earlier all biological biocompatibility studies. Whenever you give the protocol to the, of course, almost all companies outsource these studies into a particular lab, which are, you need to look at the certification of the labs before you give, whether they are CPSC approved or ALAC approved or CITISCO approved. Okay, I mean, you have in CITISCO, you have a list of companies, list of test laboratories which are approved if you go into the CITISCO site. Because though when you get your devices tested there, only the results are valid, or the test reports are valid. There are some companies where, but one called ALAC is very good. Some few of the testing, like mainly on the analytical, you have NABL accreditation, that is other are also good. Okay, so what happens here is the competency of the evaluator and documentation requirements. It clearly says that once starting from your biological evaluation plan and the protocols which you share with the laboratories, and of course the laboratory QA, I know these laboratories don't have competent persons. I have looked at a lot of people who are working in these labs. They are not so competent. They just read the rules because they are not so versatile or not good understanding the device. Okay, they go as per the textbook style, like, okay, this test we need to do. Until unless you understand the device, okay, the contact time and all that stuff, though they ask a lot of questions, but at the end you need to review it, you need to sign it out, because once the tests are started, the tests testing has started, you can't revoke back. You can't come back saying that this protocol is not good and you need to you can't restart the studies. And the worst case is after all the tests are done, you suddenly remember that this point I missed. This protocol is not fine. Actually, my contact time is much more. Okay, and I need to do some other thing, prolong the test. This is very, very difficult because I said it's a very costly affair. And second thing, though it's only once in a lifetime, it's a very costly thing and you can't come back. And as of now, you see like the rules have become very stringent on biocompatibility. If I tell you the EUMDR rules that I mean not EUMDR, USFDA, they are still much tighter than what we are discussing today. Okay, so it's better to take the help of the expert in toxicology or pharmacology who can understand, okay, both your device, okay, and understand the biocompatibility studies protocols, okay, whatever tests, because you have almost like part 1 to part 20. Okay, understand is first you need to read all the tests and should follow the protocols. Because I know, like, I mean, I can tell you, there will be a lot of standards or stringent rules in doing biocompatibility studies, not with this document, earlier also. Like how you interpret this, okay, simple cytotoxicity study, where how it is done, how it is interpreted, and suddenly you find after you get the reports, you are lying somewhere in between on the wall, standing on the wall, and you know something is wrong, and you, of course, people can get it right by manipulating it, but that's not the way because once you manipulate it, now it will in humans it will show the effect. So better get a competent person who perform evaluation competency traceability matrix. Okay, rational for omissions. If you are omitting any studies, you need to write, give the justification for why you have omitted. And of course, this we studied, it has to be linked with risk files. So earlier, like, I mean, the same guy or regulatory person who is there used to write the protocol and all this stuff for biocompatibility studies, but now you need to be more cautious, careful when you're designing the studies. Uh, Basically, at the end, what are the endpoints, study endpoints, you need to be careful uh, and include all those which you have missed earlier. Okay, and perfect biocompatibility will studies will give you a lot of weightage on your risk management. Okay, and once the risk management is complemented by biocompatibility and clinical evaluation report, there is nothing, I mean, there's no turning back. Any auditor, anyone, we can face it. Okay. So they say that you need to have a toxicological expert and all this stuff. So this we used to do basically for clinical evaluations, clinical evaluation report. This was mandatory that you need to get it signed from that particular specialist or who is the specialized, the physician or the doctor. Okay, you have hemostats, like you need a lot of people who are specialized because hemostats are used everywhere. So basically, if I have a cardiology device, I need to get a cardiologist or interventional cardiologist or a cardiac surgeon to sign that my report is right. So, which was there earlier in clinical evaluation reports or in the clinicals. Now they have moved also this thing on expertise, explanation, review from the toxicologist or a pharmacologist and an expert who need to sign this. Okay, and they also need to share their CVs, qualifications. Okay, this was very well done in clinicals by a lot of companies. And this already discussed when you omit some of the tests. Okay, you need to give justification of the omitted tests. Why not tests? omission ratio must be robust and risk-based. So this word you find even when I'm sleeping, I find this word risk-based. Okay, every test we have explicit document, scientific justification, try to export chemistry risk, generic statements. Generic, earlier we used to give all generic statements. No, they are not acceptable. Okay, I used to give a lot of statements. Earlier I am very good in giving generic statements, but now it's very much tight. Okay, expect regulators to request full reasoning, not only referring the old that's 2018 version. If your justification is weak, you may be asked to perform additional testing. So the problem with back compatibility is two things. One is very costly. Second is you can't keep on repeating the stories. So these are some of the changes. I'll share all these documents with you. I'll also share the BEP, biological evaluation plan, and the gaps. Okay, what is new we already discussed. What are the practical implications? What changed already discussed. Why this matter to NB? See, that's how I think I told your organizations first need to inform to the NB that we are going to adopt to the new standard and give a biological evaluation plan and give some timeline. By this timeline, we are going to adopt, get this test done. First thing is know what is the gap. Restructure your BEP. Okay, duration, particulate, risk, all this stuff. Extractable, leachable, get some competent evaluator. Discuss. See, never blindly believe the protocols provided by the test labs. I'm telling you, 99% there will be a lot of mistakes in it. Okay, you need to review it perfectly. Okay, place your standards one side, have understanding on your device. Okay, I mean, don't take the laboratory expert or guy, whatever name this could be, or designation it could be. Just keep him out of the loop. Once you have an understanding, discuss, okay, read the document, then you come to a conclusion. And also take his suggestion, but cross-check. So this what notified bodies may require updates on BERs, BEPs for ongoing products. For already existing products, I said you need to give a new documentation that these are our plans. They will, if this is the best smart thing we can do whenever there are some updates that are happening. Okay. You have to inform to the notified body or to the auditor that we know it first thing. We know that these changes have happened. Second thing, this is our plan. Whether it's biological or risk evaluation or risk plan or clinical PMS. First you give this is the plan as per the new requirements. Share the plan and tell in the plan you give all very structured ways that this is how we included all those in the new updates or new additions. And tell that within this time frame we will do and we are going to get it tested or do these trials in these labs. That they will be happy. But don't leave these auditors or not bodies in blind, thinking that you don't know that the changes have happened or you are not aware of it. Okay, and you don't have any plans to update your system or your studies. Okay, that basically irritates a lot of auditors or notified body. So the best smart thing is you need to be in contact with them, say, call them or visit their office, say that this thing has happened, this is the plan, this is how we are doing. If you do it or not, they'll be happy. Okay, and leave US because US is something which is much difficult to understand. I said like they always look at every device as a long-term contact. Okay, sometimes you feel interesting. because for them safety is such important. And second thing is they look at safety risk in design or safety at the design stage only. They won't prolong it to biocompatibility studies. Yes, they still want biocompatibility studies. They want to eliminate most of the risks during the design stage. Okay. And implications with India CDSCO. India CDSCO also said the same. Give your new BEP, biological evaluation plan. Okay, conduct your studies. Same like, again, I don't want to repeat it, the same scenario for any, whether it's EU notified body or CDSCO or it's US FDA. Give the plan, but it's almost 2 months this has come in. But better still, you can do it now. Okay, it's I think it's 2 months the new standard has come in. So for CDSCO or EMD or FDA, anyone you have to do this. If you have already done this, great. But again, the selection, there will be a complexity in the selection of the tests also. You know that this test has to be done. Okay, I know, like, so genotoxicity has to be done. But genotoxicity can be done in two ways. There is an in vitro method of genotoxicity, what we call reverse mutation test. Other is Ames test. Okay, teratogenicity can also be done by in vitro and on embryonic cell lines or placental cell lines. We can do the toxicity of these materials on those cell lines, not necessarily on the animal models. Why you want to kill a pregnant rabbit or rat? Okay, even if you look at CPCSC standards or if you go through them, one thing is they don't recommend going for in vivo models because there are some ethical standards or animal ethical committees which say that killing animals is wrong. Okay. And the second thing they say, if you have in vitro models, better you do and interpret what will happen in vivo. But there will be some exceptions where you need to do in vivo mandatorily, basically for performance studies, not for toxicity. And the second thing is any redundance of tests. Okay. No guidelines will tell you because you already know that the device has this toxicity or this your raw materials are same, your manufacturing process is same, if the device is the same, intended use is the same. What we call technical, biological, clinical equivalence, everything is the same. Why should you conduct biocompatibility study again? Because already the results are available. Okay, it causes redundancy of the studies. I mean, these are some which some people accept when you give justification. Like I have given justification to CDSCO so many times, quoting the standards or CPCA standards, and the standards where redundancy of the study is not necessary. Okay, because it's the same device, same results you will get. Why to kill animals? Okay, in the ethical point of view. And the second thing is they'll agree for it. If you look at the flowchart that is there in 109931 also, it also says that there is no need of doing the same test if the data is available. But they only tell you to do a comparative study of your predicated device and your device. Okay, comparative study in the sense mainly on chemical characterization. Okay, material and chemical characterization, you need to show to them all the materials, all these in my predicate and my devices are the same. Okay, and the quantity is the same. I mean, when you quantify it, or the amount of the material in that is also same. So that's one way of justification. Whatever data or the results that are there in the biological studies of this device, obviously will mimic or it will be the same like the other device. Okay, but there you need to show a lot of proof on material and chemical characterization and mostly do a lot of leachable studies, leachable and extractable studies in polar or non-polar solvents. You will have different set of solvent. Even in neutral solvent, whether it's extracting the leachable. Few tests can be done, mainly extractables, leachables, and one is chemical characterization and yeah, material and chemical characterization. Then you can give a good justification. Okay, 2-3 times I have done and I passed it. Sometimes they didn't agree. It's like saving money for the company, like. The immediate practical I told you, I have this one. I mean, this is not the original. It's uh, it's shared by some of my friends. I didn't purchase this document, but I have it. I have not hacked or pirated. It's not a pirated. Okay, I do the gap analysis like what I told. It's only 6-7 changes, but based on your device. If you tell your device, I can write your BEP. Okay. What is your evaluation plan? Okay, do the gap analysis, first get the standard, do the gap analysis, write the BEP, new BEP, structure it, reassess previous test data, look at the past test data and what the new test data you require, a new test that require. Engage with notified body, this I told you. Talk to the notified body that we know all this, what is happening. Biocompatibility, we are planning this. Show them 2-3 quotations that we already got quotations for this test and we are planning to do it. We have some financial issues, but however, we'll get it done. And give some uh, letter. If you have problem, let me know. I'm good at writing letters to these notified bodies. Okay, and include some trained relevant staff because this is a very different subject. Okay, it's not like some production or some regulatory standards. It's so for this you need to understand it's not just you're from science background. Okay, you need to be particularly from a physiological background or some pathophysiology background, toxicological background or pharmacological background. Some doctors also may not understand this because all doctors can't understand pharmacology and toxicology. Okay, and they are more relevant with the toxicology, pharmacology of the drugs than on the medical devices. So we should have a domain expert who is good in understanding medical devices. Okay, and also these studies. So this I have already spoken with you. This is new clause and what has changed, required action, whether it's urgent, immediate, because there are some which are urgent on high priority. Right, immediately you need to integrate with risk management. This is on high priority. Okay, there are which you need to do immediately. Okay, update scope of BER. You need to write a new BER. This you need to done immediately. New BER as per the changes. Update the glossary. This is more administrative. I mean, paperwork like saying that. I mean, because the terms and definitions also change here. So the glossary, like how we do in 13485 scope, we need to change. We are following 2025. Okay, the new version. Don't keep 2018. Okay, that will be a big NC if you keep 2018 in this. Okay, review it BEP and all this stuff. Integrate with the risk management. Rewrite BEP risk. Link them with 1491 section. Like I said, hazard situation found. What are the risk control? Right, cross reference. I mean, refer, link it and give cross reference to risk management. That this has been included in risk management. Okay, that document has to be very visible to the auditor or for yourself. We have linked it. This is the document. Okay, and involve the toxicologist qualified. Many people don't find it, but at least use their CV and their signatures. Particulate size, you start working on the particulate matter, what is there on your devices. Because there are different ways to measure the particulate matter because they are on the device is difficult, but you can do simple like an immersion test. Okay, a total immersion of the devices in some solvents. Okay, some organic or some inorganic. Then your particulate matter, everything will come down. Then you need to filter it. Okay, use 0.03 micro, 0.02 micron filter papers. Then you'll get everything right in vacuum oven. Then there are equipment which directly give you automated particle count and the size. Earlier, people used to count under this WBC, what you call WBC chamber counter, like what is the size and all this. But now we have ready-made. Within 10 minutes, you'll know this. This is very important. If I write a SOP on this, and you may ask me like, what is the uh, acceptable particle count or particle size. Here, what you need to do is you need to follow what exactly the particle count will be there in IV preparations. IV preparations or intravascular preparations or like injections, intravascular injections, okay, or like the saline, intravascular saline or dextrose. What is the particle count in them? It applies to us. There is a standard for intravascular devices. What is the particular count? Okay, and the size of the particles. Okay, sometimes they may ask the charge because if it is negatively charged, there is a problem like RBC are positively charged. If it is negatively charged, your particles, they may attract RBC, and once there is the particles and RBC accumulate and they form aggregates. Okay, because they attract each other, then there will be some clot or embolism. But here I don't know where they are asking. I need to look at the standard charge of the particles. Okay. Yeah, most importantly, object your biological endpoint based on the contact duration, all this stuff. And any additional test has to be done on neurological, biological, or you need to further extend your chronic toxicity study or include some teratogenicity or genotoxicity, carcinogenicity. Actually, to me, carcinogenicity, genotoxicity, and teratogenicity are both linked. If the device doesn't have carcinogenicity, I'm pretty sure it won't have genotoxicity and teratogenicity. You can rule it out. In case if it has carcinogenicity, okay, then you can go for genotoxicity because for a compound to turn from a positive genotoxicity to a positive carcinogenicity to a genotoxicity, it needs to have more contact time. Okay, for the gene to mutate to cause genotoxicity. But parallelly, I think like genotoxicity and carcinogenicity are both something like to me looks synonymous. But for temperature density is different. It has to cross the placental blood barrier in the pregnant woman or in the and it has to enter into the fetus to cause all this cause carcinogenicity or teratogenicity. But we can understand whether these extracts can cause placental blood barrier, can enter into the fetus. Okay. See, don't take my explanation as standard because I have my opinions. If the standard says we need to do, you can do. But I have my own laws. The standards be a template for 24 edition, and these are all replace endpoints, support BEP arguments, have a toxicologist or biocompatible, particulate matter. I have given all this stuff roles and responsibilities who has to do what is the timeline. I'll share this document. You can discuss with whoever is the head of this. Yeah. It is a top 10 priorities for gap critical things. So this is what I said. In biological evaluation report also, you need to write like this. What is the hazardous situation? What is the harm you can call it risk also. What is the endpoint? What is the test? What is the outcome? Generally, we don't do this in biological evaluation report. We do this more for risk management reports. Where this is the roadmap. I have my own roadmap, but I can recommend different roadmaps for you. So we'll have a short break. Okay, this is not for you. I'll share this document. If you want any help on this, on sharing the biological evaluation plans, additional tests, gap analysis, okay, or I mean, what should be the criteria for the final test reports? I will be glad to help you. Okay, just take 5 minutes break. We'll come to the real. It is 8:30. Maybe another 45 minutes. Another half an hour we'll discuss on risk management, because this I want to start. Because once I start, I begin, it's like it's half done. So I'll start it. Take 5 minutes break. Actually, break is coming. I don't know whether you need break. So by 9 o'clock, we'll have a comprehensive overview on the risk management, what we'll be discussing, what is important, why these changes have happened in risk management. Risk management also a lot of changes happened. So compared to the 2019 document, there are a lot of amendments that happened. How to do this risk management, I mean, how to identify all this. Okay, we'll discuss. Okay, don't go anywhere. I want to see the head count, including me, 7 up there. Otherwise, I'll share the document. You keep reading. I'll join. Document. No one. Yeah, before that, yeah, see, one thing on the new edition on biocompatibility studies. First thing is check whether your organization devices has any impact on this. Okay, your devices has any impact on that. Okay, if there is an impact, I think that it's better to act. And the second thing is, though it may not have impact, but I think a lot of devices will have impact on this. Mainly, I mean, on the particulate matter and other things extra. I mean, it depends upon the device, but at least have some knowledge, like you know what are the changes that have happened. I know, like, knowledge is not always wisdom, but have knowledge. And the third thing is, don't uh, look at only your devices. Look at other devices, like how it can impact uh, or your colleagues or somebody's devices, and inform to them that this could have some effect. Because once you start in a practical way of thinking or writing a biological evaluation plan as per the new guideline, you will remember it a lot. I know, like, uh, most of the people now, you won't be there in the same company till you retire. Okay? And I told to somebody that February and March are the months where there will be a lot of repositioning or change or openings that come. So don't, I mean, look at only your domain or your organization. Look at other organizations where this could have an impact. That's a better way to review. So before I go, let me show the standard. Then I'll come to that. Come to my notes. Can you see my screen? No,
Yes, sir. It is in downloads, not a document. Downloads which you mean.
now can you see?
Yes, sir.
Yeah. Actually, this is the one. Yeah, this is a new version. But this is also like includes the what I call EN ISO 14971. Just remember that EN ISO, it is different. ISO is different. EN ISO is the one which has Z annexes. Okay, I'll also tell you what this Z annexes are, but in next 10 minutes, I'll just give you an overview of it, like how it is structured. Okay. Now this application to medical devices for all medical devices from class 1 to class 3. And you know, like most of the medical device classification is done based on risk. So this is the basic document how the devices are classified. Okay, whether you take any country classification, the classification is done based on the risk, and this is the one document. That's why it is very central. Okay, like how you look, how you see in ISO 13485. Here we have scope. Okay, normative references, terms and definitions. These three are like looks common to what we see in ISO 13485. And here also think them as clause. Like fourth one also is similar, like where we look in clause 4, general requirements for quality management system. Here it is also general requirements for risk management system, that is clause 4. So in normative references, okay, terms and definitions, you need to understand a couple of things, because the terms that have been used here has a lot of impact or effect when you look into the when you write your biological evaluation plan or biological evaluation report, BEP, you need to use those terminology. Okay, this is a general like how we have class 4 quality management system general requirement. This talks of the process. Okay, the management responsibilities, competency of personnel. Okay, this is very important. Risk management plan. Okay, and the risk management file. Risk management file is a combination of your plan, your reports. Okay, anecdotes, whatever you do, whether you combine it with clinicals, how you integrate with the GSPR and all this stuff. Totally, it will become a plan. I mean, file. So first thing what you need to do is not only this, for any important standards or documents, first you need to write a plan. Okay, based on your device, what device, basically what risk class it belongs. Okay, how you will do the risk management for that particular device. You need to write total device risk plan. I will share you a format on okay how to write a risk management plan. I can share for three, like class 3, class 2, class 1. You can take the template and design as per your devices. So section 5 talks about risk analysis, how you analyze the risk. Okay, analysis process in the sense how you identify the hazards. Okay, it also gives some examples like what is an hazard, what is an hazardous situation, when a hazard encounters an hazardous situation, how can it turn into harm? Okay, it also gives examples like what could be the potential hazards, because I said like risk management, you need to look at total life cycle of the product. Okay, from your designs, manufacturing, biocompatibility, usability, all that. It will give you a lot of hazards. Okay, till your post-marketing. Okay, post-marketing or post-production is more linked back, uh, again back to the hazards. So it will, but hazardous situation is something which you need to interpret. Like how can this hazard can encounter a hazardous situation? Okay. Again, based on the device, what is the intended use of it, where it is used, what is the contact time, what is the duration, okay, and main importantly, you need to understand. See, when you look at the total device, you understand what are the critical components of the device, okay, which may enter into an hazardous situation and cause harm. Okay, it could be an environment. I think I discussed this. It could be from the patient. It could be from the physician. It could be from the operation theater or general wards. It could be from our devices. So from our devices, we need to identify what are the important weak areas for its safety and performance. Okay, and mostly the safety issues. I'll give you an examples while I come into analysis of how an hazard can become a harm in a particular environment or in a particular situation, which I some time back I also told to you. Okay, like you can look into your device from designs. You can look into your device and say that this could be a potential risk, but still I have any control over it. Okay, I can look at your validation reports and see that this could be an hazard, but still I have risk controls over it. Okay, I mean, this is what risk analysis talks about. What is the process? First, you need to look at the intended use and reasonably foreseeable misuse. Okay, it could be, I mean, that time I talked in back compatibility reuse, but here it is misuse of the device, which you foresee. There could be many foreseeable misuses which you can identify. In that situation, okay, how you will analyze the risk? Again, I am telling you, you can say that I have given IFU, I have given this training, I have given brochures, training material, all that. That is the last option. But when you come to ENISO, that option is also not there. Your IFU is not a risk control document. Okay, you can't control risk with IFU or training material or brochures. Okay, what are the reasonable foreseeable misuse? This is very important because most of the, see, when you write a risk management report, you write the risk management based on the intended use, okay, specific population, environment, all this, considering the patient, doctors, on your clinicals, okay, how your clinical reports came out. Because in your clinical reports also, there will be some safety issues. Okay, some adverse events that were occurred. Okay, if I take an example of the area where I work the most on the implantables, okay, there will be valves or catheter stents where the death rate could be 1% or the mortality could be 1% based on the clinical trials. But it's the choice of the patient. Okay, in the risk benefit, I say like there is a chance that 1% may die in 100. Now it has come down. When I'm working some 10 years back, it was 1%. Like now it has come to 0.5% within 6 months. They also give a duration within 6 months, 0.5% will have a reoccurrence of it, or he may have mortality of that. So that based on that intended use, based on our some data, we write, okay, the risk management file or the report. From the design stage or to the clinicals, we write it. But there will be some reasonably foreseeable misuse, which, I mean, we need to understand what are the misuses that it can happen. So we need to consult a lot of people inside the hospital, outside hospitals, physicians, experts to understand how this can be misused. Okay, like how in drugs, a lot of OTC, over-the-counter antibiotics are misused, and a lot of diazepam, what we call benzodiazepines, or drugs used in insomnia or anxiety or cause or misuse. Okay, so this is very, very important thing. You can work on the intended use and imagine like this could be the risk, but still I have risk in the sense hazards can storm into harm, but it has to encounter a particular environment or a situation. That's why I say not all hazards will turn into harm. Okay. In case the hazards are turning into harm because of particular hazardous situation, then you need to analyze it. Like what is the hazard situation? Okay. And can we have risk controls of it? And the most important thing is whether the risk controls you are placing, it could be 3 or 4 or 5 risk controls you have. Are the risk control effective to combat this risk? Or should you add more? Are risk controls only the way? I said hierarchically how the risk controls can be placed, how the auditors appreciate it is first risk control by inherent design. By design only you need to control the risk. Most of the risk should be controlled in design stage. Then comes your processes and procedures where you need to control, whether it's your production or your sterilization or your QC, you can have the risk control. The third is which auditors don't advise, as I said, not auditors. The standards don't advise, mainly UMDR don't advise is your reference material to the doctor or the training, your IFU training material, all that should not be considered as risk controls because they won't control the risk. Your IFU never controls the risk because we know pretty well IFU's training material brochure. This nobody reads. But a training may help good, but no physician is, except for the new medical devices, we train the doctors like this is a new device, we have modified this so and so. So instead of this pressure or instead of using this, you need to use like this. There will be training to the rest of the physicians also when new devices or modified devices come. But for already existing marketing devices, nobody will care the IFU or your brochures or your training notes. Okay, your inclusion, exclusion, because they have a patient and he has to attend. Okay, so that's why I'm saying like when we discussed about copper, we said that what is the effectiveness of copper? If there is no effectiveness of copper, it will reoccur. But the same complaint reoccur if your copper is not effective or your root cause is not addressing the relevant problem. Your copper becomes ineffective. So here your risk controls may also become ineffective if they are not able to control the harm or the risk. Then you need to relook at your hazardous situation. What is the situation? Not the hazards. You can trace a lot of hazards. You can write hundreds of hazards. But what is the hazardous situation is the key. Even when you do a copper, when you work on the root cause analysis, this is very much linked. Hazardous situation is very much linked to the root cause analysis. Okay. Then what is the risk control you will have? If this is not effective, what is the risk control? You will go back to the design or change your processes, or you'll have more effective risk controls. Okay. Though you may have risk controls, what next? Okay. Are you able to justify the risk? Okay, that is what is the severity of the risk? Okay, this is risk, and then we come to risk controls. Risk control option analysis. How we will analyze this? This is able to control the risk. Okay, risk control measures. Then you'll come to after I control the risk also through these processes to this risk control. You can pick up a lot of risk controls. Like biocompatibility is one risk control. Your biocompatibility reports, sterilization reports, your QC report, internal audit. There will be a lot of information you can use that as risk controls. but still you feel that, okay, most of them say risk is acceptable, but nobody will say there is no risk. Okay, then you need to look at what is the residual risk. Still it has a residual risk, but what we do generally is we will show residual risk at a very minimum level. Like, though it may occur, there could be a minor bleeding, which can uh, be subsided, or which it can be worked out, or there could be some minor detachment which may not affect the patient. So residual risk will write in such a fashion that it won't have a direct impact, okay, on the patient safety. I mean, that's the traditional way of writing. But still, the question here is, can you bring the residual risk still down? Again, I said it includes a lot of things on economic considerations, redesigns, and all this stuff. Okay, this will be asked when you do the risk benefit analysis. People call it as benefit risk analysis, but I am very used to calling, since I started working on these documents on risk management, I call it as risk benefit analysis. Then even the standard says benefit risk analysis. I use the other way. I'm used to it. So your residual risk evaluation, then benefit risk analysis. This is another very important point, risk arising from risk control measures. You take risk controls. Suppose you have adopted some risk controls, maybe from design you have adopted, that's what example I told. But while you adopt the risk controls, there could be some risk that may arise because of your risk controls. Okay, I'll tell you when we go in depth some of the classical examples, when some of the risk controls will add additional risks. Okay, then what is the completeness of the risk control? Of course, risk controls can't be, but still you need to write this is the completeness what I can do for the risk control. Okay, and then overall residual risk, like you need to add up everything. What is the residual risk after doing all this stuff on analysis, hazard situation risk, risk evaluation, risk analysis, risk control, risk mitigation. Okay, severity, occurrence. I'll tell you, go in deep on how to write the severity and the occurrence. Okay, there's a very classic document which is not there in this 14971, but there is one document which I read long back, how to understand the occurrence and risk. Okay. And you need to sum up all your residual risk here, overall residual risk. Okay. Then you need to say, this is one area. After doing the overall residual risk, you need to link this to your clinicals. Where you link this to your clinicals? Because this residual risk will be in parallel proportional to your adverse events or complaints or serious adverse events that occur of your device or the because of residual risk. You find absolute correlation. Okay, you tell me any device, I'll tell you how your residual risks are linked to the adverse events or serious adverse events or complaints or some, how we can link it up. So once you link your residual risks with adverse events or complaints, how you need to translate them to the patient. How to tell the doctor and patient that I have residual risk. This residual risk will further propagate into some adverse events, some complaints, okay, or some serious adverse events, like I was talking on a 0.5% mortality rate. we need to tell it that the device has this, there is a mortality that can happen. Because I have conducted, I mean, historical data says that this device will have, and my data also shows 0.5% mortality or morbidity in next 6 months you will have. Okay, I'm talking about high risk and also class B, class 2B, and class 3. Okay, I mean, that's why I look at, you need to look at your device classification before you write the risk management file. Because for class 1, there won't be any risk. I'll show it for class 1, how, what are the things that are required. Class 2 and class 3, what are the things that are required. So here what I'm trying to make you connect is that residual risks are directly linked to your clinical events, adverse events, or serious or something like that. And how you convey this to the doctor or to the patient. Patient don't read anything. But how effectively you can communicate is that you mention that in the IFU. How you mention it in IFU? Okay, either you write them as warnings or precautions or contraindications. So these three categories will directly tell you what are the serious adverse events or very serious adverse event. You can classify this as you like. Okay, or compliance. And these are basically where from these events arise, basically from the residual risk. So now you need to understand three things here. One is the environment, the hazardous situation. Hazardous situation is basically linked to the usability engineering or human device interface. You have one document sitting there in the usability engineering, how to use it, and what are the human device interfaces, whether the human may be the doctor or the patient and the device. So it's more like a triangle. Okay, you have usability engineering. Then you have in risk management where you have residual risk. And these residual risks are directly proportional or linked with your clinical adverse events or events. Okay, and these adverse events has to be conveyed. They come in the form of an IFU. In IFU, how you mention them as warning, precautions, or contraindications? I think I discussed somewhere. Contraindications are something which absolutely advise not to use the device in that population or in the patient. These are totally contraindicated because again you need to do some reverse engineering on that. Why I say that those those in contraindications should not use the device. One is you come back, okay, to the adverse events, or it could be a serious adverse event. Like if I use them in children and pregnant women and geriatric patient or above 60 or somebody sensitive to some of the things which I have observed in my design, which I have observed in my biocompatibility studies. I mean, this has the leeches or this leeches nickel is sensitive to him, or I know like this may come harm to the particular patient who have this blood group or hemorrhage. Okay, this will give it because these are coming from the clinical events. And these clinical events are coming from the residual risk because you know what are the residual risks. So you have one wherein your usability engineering is coming here. Okay. And you have the risk management which is sitting in the center. And you have your clinical evaluation reports. And from all these three, you derive at the IFU. Okay. All of them are interconnected. Okay. After the device is used, then you have a PMS, what we call post-production activities, or post-production in the sense is more marketing. In the marketing, you discover maybe you are doing a small registry or you are doing a PMCL. But in the real world, you are looking, that's why I said it's post-marketing is basically the real world, not the controlled clinical trials where you have a particular subset or group or cohort of the patient, where there will be a lot of benefits, performance will be super, and you don't identify any risk because it is a control group. But when you go to the real world, you'll have a lot of variables, variety of patients, comorbidities, different age groups. Okay, you find everything. But your device is not tested in all the population, all the patients. Okay, the physiology varies, the pathophysiology varies, the anatomy varies, everything varies from patient to patient. Their ADMA, total kinetics vary, the pharmacodynamics vary. You take any vitals, functions vary. But this you have not tested when you are doing your clinicals. So once it goes into the real world, then you will have some more adverse events coming in. Okay, it's patient linked or it could be doctor linked or some hospital linked. Then you need to trace it back to your risk management. So these are the new risks, new adverse events coming. Okay, new harm coming. Okay, in a particular hazarded situation, this is a situation, this is can I have more risk controls for this? Okay, your PMS is providing because it is the input for your risk management. When I say PMS, there are a lot of things like your customer feedbacks, your complaints, okay, your predicative device complaints, because that's what we need to look at. Trend analysis also includes what are the risks and benefits of your predicative. You should have a one eye on how your predicative or the best device is working. Whether they're having similar complaints like my device, or they don't have complaints, but I am having complaints, or your device is having new complaints which is not seen with other devices. Because this happened, I know, like when the stunt were introduced first time in 2010, okay, nobody knows that post marketing only people could understand there are so much of problems, okay, with the stunts, metallic stunts. Okay, and that are so threatening. There are life-threatening feedbacks. Okay, both on the drug-eluting stents and also on the metallic stents. So there were a lot of changes that happened next 5 years, 10 years. Now we have a better system. Even India is producing good quality implants like heart valves. Even there was issue with the heart valves when used in pediatrics. Okay, a lot of deaths happen, and not in pediatrics, even in neonates, 6 months babies, like when you have pulmonary valves not opening up or dysfunctional pulmonary valves. So what I'm trying to say is PMS, you should look very carefully in the objective of the risk management. This will give you inputs or what are the new risks encountered which you have not documented in your risk management. That you need to add. And you also understand what is the situation that caused the risk. That's why I say you need to study complaints, okay, in all dimensions. Because there you will find why that hazardous situation happened. And the hazard has converted into an harm. And the best company, what it does when it has the PMS that is coming and it includes that hazard and understand the hazard situation, immediately it will place a risk control. Risk control could be come in any form. Okay. The toughest one is on the redesign of the redesigning your device or some change in the procedures, processor, or the best case, what I see is having additional QC checks. But again, I say additional QC checks or risk control should not add another risk. Okay. So this is the completeness of this. I talked about rescue. How you interconnect all this, I'm trying to make you understand. Okay. From the residual risk, how you connect the clinical events, from the clinical events, how you project it as a labeling. And when I say this device is better than my device, when we say that, when you have less residual risk, I mean, I'm coming directly to the first point. You need to control your residual risk as low as possible. That's what the document says. As low as possible, not as low as reasonably possible. It is not allowed. It is allowed. As low as possible. So when you have as low as possible, then your adverse events come down. Then you'll have less precautions, warnings. Of course, the contraindications may also reduce, but more or less, I think contraindications will be the same. Okay, then that will change your IFP, and that will have more acceptability with the doctor or the patient when you have this checks on risk control that are newly added, or the hazards that are newly added, and you know it is going to convert into a risk, then you immediately place the risk control or reduce the risk. Okay, acceptability or residual risk, you try to reduce it, or you have a risk control. Then obviously this, you'll see a pattern in all these guys, I'm telling you. When you see a pattern in all this, you really appreciate it. Okay, mainly people who are in class 2A also, class 2B and class 3 devices. Okay, and that's why I say that risk management is the one which holds, okay, all of these things together, and at the end of the day, it delivers a good device or a good performance device in the terms of its benefit and also less risk. Okay, this is what I said. Post-production activities, we need to do it. I mean, this is mandatory. If you look at UEMDR, they give, I mean, that's what the new thing that happened in UEMDR 2017 compared to MDD. Okay, medical directive, the old document is they have emphasized more on clinicals, clinical evaluation. They said that, you know, the main change that happened to me between the old MDD and MDR, new MDR, is more stress on the clinicals. Okay, they said very clearly, don't give me the data of your predicate. Okay, don't do literature survey and say that my device and your device are same, we are close, we are twins, we are friends, we are neighbors. So my device will be same, like that is not accepted. It's not at all accepted now in UMDA. You need to give your clinical, okay, what we call more on your device clinical reports. Your device, not your neighbor's device or your predicated device. Give more data on your devices when it is used in a particular set of patients for that intended use. What are the clinical outcomes? So that is the worry with a lot of the Indian companies, because they can't do the clinicals, which is very costly. Again, it was relaxed. Say that, okay, don't do a PMCF. PMCF is again a control trial, and you can do that and it costs a lot. Okay, you can do it prospectively, but you have already sold thousands of products, devices. So they said you get the retrospective data of the devices, what you have already sold. Okay, whether it's procedural success or performance or clinical success. Get the retrospective data mainly for class 3 and class 2B devices. Okay, I mean, that was the turning point. And the second one is they said very hard emphasis on post-marketing surveillance. Okay, every device has to be tracked. I mean, PMS includes a lot of things like your periodic safety update reports, PMCFs, feedbacks, complaints, CAPA. your trend analysis, and the real world data, like what's happening with same devices or similar device like you need to collect the data open and show that these devices have this problem, my devices have this. It's not for justification. It's to understand how the same similar devices are working. Of course, we need to look into the patient population. It's not like uh, I said, like the other situations may vary. When they vary, obviously it may turn into harm. I think I also discussed how to, I mean, at various stages you can control residual risk, okay, by having a high consistency data in your production, okay, or in your QC, like I was discussing, maybe next class I'll discuss what we call on the thresholds or on the standard deviations you have. Okay, this is talks about all this. There's a lot of it's 46 pages only. You can read it, but for your comfort, what I have done is I have separated all these sections, okay, and made separate notes clearly explaining this and what are the options we have which are not provided here. Through my practical experience, I'll tell you how we can work out on this, how we can use justifications, okay, how we can link up so many things. Like say you need to link all of this with GSPR. Okay, general safety precautions require, sorry, general safety performance requirements. Either it's earlier it was MDD, it's only EP, essential EPR, like essential principle. Now they have extended it. It's like almost 42 pages. Okay, it's like total abstract. If you read the 42 pages of GSPR, you'll understand everything. No need of looking at other documents. So I'll also tell you how to link this risk management with GSPR. Okay, how to link it with usability, how to link it with clinicals, how to link it with biologicals. End of the day, how you need to look at your trend analysis, your PMS, and connect back to them, connect back to risk management. And to sum it up, how your IFU will come out. Okay. See, this is something which we need to systematically understand and need to be, I mean, why you enjoy this is there's nothing like need to memorize it. Like how 13485, it's more on practice you'll understand, or it's more on need to some things related to memory, but it is purely something related to your logic, your rational, and how smart you are in understanding this, how you can link it up, lot of things. Of course, the device knowledge is necessary, intended use is necessary, how the device is used is necessary. Okay, and end of the day, what are these hazards, hazard situation, risk controls, analysis of risk, evaluation of risk, how you calculate this? Can we quantify this or just structure it in sentences? Okay, anything quantified is great. Yeah, there is a way to quantify a lot of things. What are these residual risks? And at the end, there will be one column where we write the risk is acceptable. Okay. Generally, I'm also a bit confused. I mean, that's the reason why there is a lot of difference between the old document and the new document. That's 2019 and 2007. The old is 2007, I think, 7 or 13. Because people, though it has risk, they say that this risk is acceptable. It has so much risk. They kept some risk control. It has so much of risk. In the end of the column, they write it as risk is absent. How can you accept that risk? So manufacturers started using this so leniently and saying that this risk is acceptable. It can't be acceptable. So that is the reason why this 2019 document edition came in, which said that it should be as low as possible. Check the effectiveness of your risk controls. If necessary, add more. or you go back to the design, show me more in clinical evidence, link it to PMS. PMS has to be rerouted to risk management. So this is the complete story on risk management, how I view it. It's great. Like, so next class, of course, we can discuss these lengthy sentences, and I don't like this kind of paragraphs. Okay, it has to be very precise. But one thing I can tell you is like, this only I rounded off. See how important it will be. Like every verb has a specific meaning. Shall, should, may, can, must. Shall means it is mandatory. Should means it is recommended but not mandatory. May is your choice. Okay. So like that, we'll discuss next class. And thanks for your time. Have a great Sunday. I think I lost my throat. Actually, I have toothache. See, in early 40s only, I'm having so many problems. Of course, I'm operated in all parts of my body. In early 40s, I had my brain surgery, I had angioplasty, I had multiple bone fractures. Actually, I'm born very weak. Okay, guys, take care. Have a great Sunday. See you next week. In the meantime, if you have any questions in it, I'll be sharing a lot of information. Spend half an hour or 1 hour in 24 in the next 6 days and go through it. Because I want this not really 13485, but I want this 14971 everyone to understand and link it up. I said again, I'm repeating, if you are good, that is great. If you are experts in 14971, I think sky is the limit for your career. Okay. Thank you. Take care. Bye.
Take care,
Thank
sir.
you. Good night, everybody.
Bye bye.